Thank you for the kind invitation. I'm Ying from the Johns Hopkins University. I'm very excited to share our OGM data in soft tissue sarcoma with you. Bone and soft tissue tumors can occur at different body sites and have complex histological features as shown here. Those tumors are commonly classified according to the WHO classification, which integrates clinical radiology, morphology, immunophenotypic, cytogenetics, and molecular genetics findings. Comprehensive detection of the gene fusions, structure variations, and rearrangements is very important for those tumors. We performed OGM on 201 soft tissue sarcomas and compared the results to standard-of-care genetic testings, including karyotyping, conventional chromosome analysis, and fluorescence in situ hybridization FISH, and RNA sequence-based gene fusion assay, and MGS-based deep sequencing. We successfully performed OGM on all 201 soft tissue tumor specimens. In total, we detected more than 22,000 structure variants SVs and the copy number variants CNVs. After removing chronogenesis-related events, about 5,000 structure variants and copy number variants remained. On average, each sample showed around 21 structure and copy number variants. The most common variants, including interchromosomal translocations, intrachromosomal fusions, deletions, gain and duplications, amplifications, inversions, and insertions. To focus on pathogenic findings, we analyzed the data using a customer panel of over 1,000 cancer-related genes, including oncogenes, tumor-suppressed genes, and no fusion genes in soft tissue tumors. With this approach, we identified an average of the 4 structure variants and 7 copy number variants per specimen. 50 cases, or 27%, showed features consistent with chronogenesis, which represents recurrent, huge genomic events involving large-scale chromosomal rearrangements. Those cases demonstrate recurrent patterns of oncogene gains and tumor-suppressed genes losses, with an average of 204 genomic variants calls per sample. Overall, those results highlight how OGM provides a comprehensive view of the genomic landscaping in soft tissue tumors. This figure shows a comparison between OGM and traditional chromosome analysis karyotyping. In our study, all pathogenic markers identified by standard karyotyping testing was also detected by the OGM, showing 100% concordance. In other words, OGM correctly identified the same pathogenic structure and copy number variants, resulting in 100% specificity. Interestingly, the OGM detected about 50% more pathogenic genomic abnormalities than karyotyping. This was most obvious in cases where the karyotyping was normal or unsuccessful. Those findings highlight the improvement sensitivity of the OGM for identifying complex genomic alterations in soft tissue sarcoma. OGM identified pathogenic structure variants in more than 70% of the cases that had normal karyotyping results. So in the cases with normal karyotype result by the cancer cells, so either the tumor cells are not dividing in the in vitro culture environment, or the cancer cells are dividing but has a normal karyotype, which has the cryptical rearrangement or structure variants cannot be detected by the karyotyping or chromosome analysis. So, for example, this is a 7-year-old boy with shoulder mass, and histology showed the blue cells, small blue cell staining, and a normal karyotyping, including normal chromosomes 7 and 22. OGM revealed a cryptical 7 and 22 translocation, leading to the EWSR1 ETV1 fusion. So this fusion is pathogenic for this patient, and which cannot be detected by the chromosome analysis. This case is a sarcoma with a normal chromosome analysis. OGM revealed a complex 20 and 22 translocation, resulting in a EWSR1 MFATC2 gene fusion, which is pathogenic for this patient. On retrospective review of the karyotyping, 20 and 22 translocation was identified in 1 out of the 20 metaphase cell analyzed. But however, 1 out of the 20 metaphase analyzed by the karyotype was not reported because it was interpreted as non-clonal, which is thinks this is could be the artifact. So OGM successfully detected pathogenic and important structure and copy number variants in nearly half of the specimens in this study, even for the samples with field karyotyping. So for example, in this unmasked specimen, OGM identified an SRF rearrangement, which was further confirmed by other methods such as RNA sequencing testing, which led to a revision of the original diagnosis from the soft tissue leiomyoma to the cellular myofibroma. This case highlights how OGM can be very important for challenging the disease entities. OGM is highly effective for detecting complex genomic rearrangement. So this is a 14-year-old specimen from the leg mass, and the patient has a recurrent sarcoma and complicated karyotyping as shown here. So the patient had a 4-way translocation between chromosome 5 to 12, 12 to 22, and 22 to another 5, and also have abnormal chromosome 3 and abnormal chromosome 16. And OGM revealed additional genes involved in this complicated gene rearrangement. It's a 6-way translocations by the OGM. So from chromosome 5 to 12, 12 to 22, 22 to another 5 to the 13, and 13 to 16, and 16 to 5. So OGM reveals additional genes involved in this 6-way translocation. Some of them may help identify new therapeutic targets. So OGM also are important to detect pathogenic variants in the cases with abnormal karyotypes. So this case, the patient had 2 cells by the karyotype has 2 cells had 1 and 8 translocation, and the 2 cells had 1 and 8 translocation, additional abnormality of the abnormal chromosome X and abnormal chromosome 2. So the OGM revealed a 3 and 16 translocation leading to the VGLL3 and CHD9 gene fusion, which is pathogenic for this disease. So when we did a retrospective review of the karyotyping analysis, we realized the 3 and 16 translocation was observed in 1 out of the 20 cells analyzed, but was not reported, and because was thought is an artifact by the culture, and so which means sometimes even you have abnormal result by the karyotype, and sometimes you also could miss the pathogenic changes. This study also highlights the potential of the OGM to detect complex genomic alterations. Coronal angenesis events is very common in this study, occurred in 27% of the cases analyzed. There are different mechanisms can lead to the coronal angenesis events, including coronal trypsis, coronal angiosynthesis, and coronophylaxis. In our study, about 1/4 of the cases showed coronal angenesis with chromosomes 8 and 16 and 17 most frequently involved in osteosarcoma. Some bone and soft tissue tumors do not respond well to standard treatments, highlighting the needs for alternative therapeutic approaches. Understanding the role of the coronal angenesis may provide insights into tumor development and help identify new potential therapeutic targets. OGM reveals additional biomarkers and genomic alterations that could impact treatment decisions. So, for example, this is the chest wall mass specimen, and the patient had malignant peripheral nerve cell tumor, MPMST, and with a complex karyotype near tetraploid and many chromosomal abnormalities. And this tumor shows a more complex cancer genome compared to another MPMST cases in this study. So this patient, each red line is a translocation, has a coronal angenesis events, and also genome-wide copy number show the homozygous deletion of the 9p, including the CDK2N and 2B. And the other case has a few changes and a few CMVs. So this study highlights the genomic heterogeneity among MPMSTs. In addition, targeted therapy such as the TRK inhibitors could become potential target treatment for the cases with CDK2 and 2B alterations. So in summary, OGM had excellent concordance with standard of the care genetic testing, including chromosome analysis, karyotypings, and FISH and gene fusion assays on soft tissue sarcomas. OGM revealed 50% more pathogenic alterations compared to the standard karyotyping. OGM revealed genome-wide structure and copy number variants, some of which may be related to novel biomarker among various soft tissue sarcomas. So I would like to thank the Hopkins Clinical Cytogenetics Lab who did this work. And GH Cytogenetics Lab belongs to the big the molecular pathology division. I will end my talk here. Thank you.